Good morning, or afternoon, or whatever. I'm Mr. Dillon from NorthBay, and today I want to talk to you about Mendelian Genetics.
Who was this Mendel guy? Well, Gregor Mendel was an Austrian monk
Who developed our understanding of genetics and inheritance back in the 1800s. Mendel bred a lot of pea plants together, kind of like these ones, and found that they had heritable elements, or as we call them today, genes.
He also determined that each offspring only receives one heritable trait from each parent, and that these heritable traits or genes that are received do not affect the other genes that are inherited. And he learned that some genes have variations called alleles. And these variations in genotype within the pea plant were either dominant or recessive. Genotypes are the genes or heritable elements that determine the observable physical characteristics that we call a phenotype for an organism, like color and shape. So basically what Mendel did was he crossbred pea plants that had different traits to figure out which were dominant and recessive. And then he mapped all of that out using similar charts to Punnett Squares, all to determine the patterns of inheritance for his little pea dudes.
A Punnett square is a chart that allows us to estimate the percentage of each genotype that will occur in the next generation and its phenotypes. By observing the phenotypes that appear after each generation, we can determine which alleles are dominant and if the parents are homozygous or heterozygous for the traits. A homozygous parent carries only dominant or recessive alleles, whereas a heterozygous parent carries both dominant and recessive alleles. In Mendel's experiment, a process called simple inheritance was occurring, wherein only one gene dictates the phenotype, and there are only two alleles or variations of the phenotype. So we just looked at pea plant color and diagrammed for the next generation. If we cross homozygous dominant and recessive parents, we would expect to see heterozygous dominant offspring, meaning they express the yellow phenotype but also carry that green recessive trait.
If we then cross these heterozygous yellow pea plants, or a plant that carries both dominant and recessive traits, we would estimate that the offspring would be 25% yellow homozygous, 50% heterozygous yellow, and 25% homozygous green, the recessive trait. This 1 to 2 to 1 ratio is common in these test crosses and allows us to figure out if an organism is homozygous or heterozygous for a specific trait.
My guy Mendel chose a great study species. A lot of the phenotypes in pea plants are determined by simple inheritance. In other organisms, it gets a lot more complex, and the inheritance patterns are more complicated. For example, some phenotypes are sex-linked, like colorblindness in humans, which means that the gene that determines if you have the trait or not is on either the X or the Y chromosome. And other phenotypes are based on more than one gene. For example, a lot of human and animal physical characteristics are determined by multiple interacting genes.
My friend Jen and her sister Kira at first glance look really similar. They are both blonde with blue eyes and fair skin. They share 50% of the same DNA, and yet they are distinct and unique. You might think that straight versus curly hair is an example of simple inheritance, but when we look at their parents, we see that neither have curly hair.
We would need to dig back further to determine where Kira gets her curly hair from, because scientists have discovered that hair follicle shape and pigments are determined by multiple genes. But there are several phenotypes that do follow the pattern of simple inheritance and can be easily observed in humans.
You may be able to see them in your family. For example, hitchhiker's thumb, widow's peaks, cleft chins, freckles, dimples, and attached earlobes.
Other traits that follow the pattern of simple inheritance are things like having wet earwax,
being prone to acne, and even being lactose intolerant. Nowadays, there are even tests that help you determine what parts of your DNA influence the attributes that make you unique. You can explore all these different attributes that make you unique, like your skin, eye, and hair color, as well as iris pattern, and much more. These scientific advancements are truly amazing. So look up some of these traits and talk with your family and friends about it. See if any of these traits and attributes make you even more unique. Discuss the importance of differences that make us unique. It's thanks to this diversity in mind, body, and character that makes the world that we live in a dynamic and interesting place. So recognize the similarities and the differences, and celebrate the uniqueness that allows us to fill our own specific niches. I'm Mr. Dillon from NorthBay, and I hope that you have a great day.